Okay, so down to business. So it's really great to have Garry Nolan here to speak today. So Garry is the Rachford and Carlota A. Harris Professor at the University of at Stanford University School of Medicine and Cancer Institute. Garry's lab for many years has been interested in hematopoiesis, cancer, leukemias, autoimmunity, and inflammation, etc. But I think Garry is also very well known for really being a leading pioneer and innovator of new technologies. Okay, and so for example, I think well before most of us were thinking about and doing single cell RNA-seq, Garry for many years was pioneering single cell methods based upon mass cytometry. Okay, so specifically CyTOF using mass cytometry for multiplexed single cell analysis of protein expression. Okay. And in recognition of that, Garry has had many awards for his innovations. So, for example, the Teal Innovator Award, and also an award in 2011 from Nature Publishing Group for outstanding research, specifically in recognition for his work on CyTOF as applied to the hematopoietic system, I think. So, as I say, a long-standing innovator, and most recently, okay, in line with the interests of this meeting, okay. So obviously here we're we're trying to learn all about spatial omics, single-cell spatial omics approaches, and so Garry has developed a technology called codex, which is multiplex immunofluorescence imaging. Obviously at the single cell level in tissues. And I think, it's really great to have Garry present at this forum, because we've heard a lot about RNA-based technologies. And so codex is, obviously a protein complement to all of that. So really nice to have you come here and speak today, Garry. Thanks.
Great. Well, thank you very much for the invitation. I'm sorry I can't be there in person, but next time, hopefully soon. I'm going to talk today about how do tissues arrange themselves, right? And what are the building blocks? And the flip side of it is, does this actually represent a new set of targets for therapies? Although at the end of the day, it will be aiming things at molecules inside the cell or on the surface of the cell. How we find those targets is important. And I think as I go through this, you'll start to see that there actually are new classes of targets that we can start to elucidate out of how tissues structure themselves. And I'm going to take it from the standpoint of something that I think you already understand, which is inside of a cell, proteins come together, either one or two proteins at a time, or they form larger complexes, supramolecular complexes. But we know that the proteins don't stay there very often. They often will partake in multiple different signaling complexes, and they'll move from one place to another. So proteins come together for a purpose, they pass a signal, and then they move on to do something else. So that concept actually works at the next scale up, especially in the immune system, where cells will come from one place in the body, head to another, will pick up, say, an antigen, they'll move back into the lymph node, they'll talk to some other cells, groupings of cells, they'll accomplish some abstract purpose, most of which we frankly don't necessarily even understand why 4, 5, 6, or 7 cells come together at a given point in time. But they do so at least transiently, and then they go off and do something else. So tissue imaging offers an opportunity to start to collate the information and then relate it to outcomes or post-perturbations, right? So the first step of this, as in any of these kinds of technologies, is, well, make it work, obviously, but then start an atlas, right? And then start seeing if certain things show up more than you would expect by statistical chance, and then try to relate it to particular outcomes or observations in the tissue that you're studying. So it's really the architecture as created and defined by the organization of niches and the intercellular relationships and how we think about that. So as was mentioned, my lab's been involved in developing a number of technologies. CyTOF, previous to this, you've probably heard of the 293-based T-cell work, something that Warren Parr and I developed way back when we were both postdocs with David Baltimore, and then PhosphoFlow. PhosphoFlow, frankly, was what led us to CyTOF because we needed more parameters. Then we got basically burned a number of times by reviewers who said, well, gee, you just took that cancer apart down to the single cell level and you left all the context behind. How dare you try to make a conclusion? So then we moved on to create Codex and MIBI, two imaging technologies that basically accomplish more or less the same thing by different routes. Here's the MIBI. This is the atomic imaging thing I was talking about before. And then another technology that's come out and you'll be seeing soon is a split pool approach to actually replace CyTOF. Don't buy a $600,000 instrument when you can do everything by split pool. And this is from a patent that we submitted way back in 2010. So I have many commercial conflicts. The whole purpose behind actually making the commercial side of things is to basically get this out of the lab and into other people's hands. And then they'll do all the hard work of the quality control, as if you overheard previous discussion, that's where things need to go. Okay, so let's begin right to Codex. Okay, so this is a multiplex annealing approach. We published the first of it in Cell now 3 years ago. We had another paper last year I'll talk about in a minute in Cell using it for cancer. So you stain with anywhere from 50 to 120 antibodies. These are, of course, all validated antibodies. We cross-check it with pathologists, et cetera, to make sure that we're not seeing things. You cross-link those to cells all at once. And each of those antibodies has on it a unique DNA tag. And it's that DNA tag then that we use to anneal up to actually 5 different fluorophores at some of the upper ends of the tech we have going, where the oligonucleotide directs it to the antibody of interest. You do the imaging at whatever size of the scope, the tissue that you need, at speed and resolution that you require. That's actually the slow part. And then you de-anneal, right? You just denature those oligonucleotides and you come in with another set and you repeat this. Actually, the chemistry only takes about 15 minutes. It really is the imaging that takes the majority of the time. So you're getting these iterative images, you build them up, you align them. There's a lot of computational work that goes into aligning all of them to make sure that you've got all the kinds of registration that you want. And then the fun begins. Now, this is the instrument. You guys actually will have one of these. The first instrument that we built was literally built out of Lego blocks. The guy who built it had a couple of kids at his house, his home, and they had Legos, and he used that as the platform to develop the original robot. And so why do it, right? What's better about multiplex immunohistochemistry versus, say, individual markers, right? So PD-L1, immunohistochemistry marker that you guys probably all know about. It's an immune checkpoint marker compared to a multiplex immunohistochemistry. More data gives you a better AUC. It's better than the best of the expression profiling. This is from a compilation of multiple clinical programs that were done and tumor mutation burden. So it can be better than just individual markers alone. And of course, there's a lot of other things you can do with it, especially around the immune system and outcomes and understanding of mechanism. So here's a short list of the antibodies that we have working in my lab. In fact, it's much larger than this these days. But you can see we generally break it down into all of the immune system markers, tissue-specific markers, as well as you'll see scattered throughout here, a lot of activation markers. Not on this list are many of the phosphoproteins that we use. Here's just a short number of them. Here's the STAT proteins, et cetera. So basically anything inside the cell, inside of the nucleus is fair game. If you've seen it published in a paper somewhere, it's most likely that we can adapt it to the FFPE or fresh frozen techniques that we've got going in the lab. So I'm going to jump right into the example that we just published last year of colorectal cancer and how we applied codex to this. So this is the work of Christian Sturch and Darcy Phillips, 2 clinical fellows in the lab. And so we started with 750 colorectal patients where we had fully clinically annotated material from all of them. We broke it down to stage 3, 4 extreme cases. And from these, we chose 2 extremes of those extremes, right? What's called a Crohn's-like reaction or a diffuse inflammatory infiltrate. These are the patients, the CLRs that do better. I'll show what they're like in a minute. And the DIIs do worse. So there's a spectrum, of course, between here, but these are the 2 extremes that were of use in this particular study. So we had pathologists look at each of the slides, each of the blocks, choose the tumor immune interface and take a punch at that interface. In fact, we took 4 from each patient to get a decent sampling of the tumor microenvironment, put them all on microarrays and ran them all, right? So here's just the H&E staining of a Crohn's-like reaction. What it actually is, the CLRs, is actually in here, as you'll see in a minute. This is a B cell follicle. There's a number of cancers that have B cell follicles that set up within the tumor itself. You can think of it as a forward army camp that's setting up within the tumor. Makes sense. Why go all the way back and forth to a lymph node when you can basically bring the lymph node to the cancer? These are the ones that do better, right? And then over here looks like basically an ice cream swirl in a way. This is the diffuse inflammatory infiltrate. Some cancer and stroma within here. You can see the immune system cells scattered out, the smaller ones. And these are the patients who, as I said, do worse. And here's the Kaplan-Meier curve showing you the outcomes for those kinds of patients. All right, so let's start looking at some of the markers. So here are then, of the 50 or so markers that we used, here are 7 of the top level markers that call out the various cell types, basically Tregs, macrophages, B cells, et cetera. And you can see this is that follicle that I showed you before. That's the germinal center found, but that is inside, or something that looks like a germinal center that's inside the tumor itself, right? And then over here on the right, you don't see any of those more organized structures. And that actually, that lack of organization that we thought was lack of organization, at least to the naked eye, ends up being not the case, as you'll see as I proceed here. So we segment the cells computationally, figure out where they are, make assignments of which cell is what type based on the marker expression profiles. This is all now automated post data acquisition. And you can see again, here's the follicle, and you can see that there's obviously other kinds of cells scattered throughout all of this. And then over here on the right, the DIIs. And you'd look at this and you'd think, oh, well, different kinds of cell types, different kinds of neighborhoods. And as it turns out, that's wrong at a grander scale. So rather than looking at all of the individual cells, we developed this concept of cellular neighborhoods, where what we do is we take an index cell, we look at let's say it's this cell. I don't know if maybe I'll turn on my the pointer and then you can see it. So you'll take an index cell here, that pinkish cell here. We'll go anywhere from 10 to 20 cell radii out, count all of the cells that are within that area, just make a notation of that, go to the next cell throughout the whole system. And then we'll look for groupings of cells, not the particular order of the cells, just within that window, what are those cell types, and we'll call it and demarcate them as a neighborhood. Now, we can go 5 cell types out, 10 cell types out, 20 cell types out. So it's somewhere between 5 and 10 that you end up getting basically the same thing above. And if you go too many cell types out, you're basically looking at a whole tissue and you don't see any distinctiveness or not. So 10 to 20 cells are what we usually use. And then you break them down into then what we call cellular neighborhoods. And so here are the cellular neighborhoods at a pull-out large scale. So here's what that looks like without the cellular neighborhoods, and now we're breaking them down into about 10 or so different cellular neighborhoods, where actually the majority of the cells that are out here, or at least we characterize them as NK cells, right? And then here, within here are the B cells and other little neighborhood types. Each of those little dots, or the Voronoi that you see here, that's a cell type. The color is the neighborhood. The surprise was that if you actually look in the DIIs, you don't have different neighborhoods. You just have different arrangements of the same neighborhoods and different frequencies of the neighborhoods as to what you saw in the CLRs. So there's not a difference between the DIIs and the CLRs in terms of the kinds of neighborhoods that are formed, but in the DIIs, and as we've seen in several cancers, the worse the cancer, the more fragmented the neighborhoods tend to be. And especially more fragmented are the immune system containing neighborhoods. And it seems, just as a general takeaway, that worse off cancers seem to make a strong effort to actually disrupt the immune system and prevent it from coming together and coalescing into a functional unit. And I'll give you some more examples of that in a minute. So rather than repeat the whole paper, because it is already published, because there's a lot of work I want to talk about, I'm just going to kind of jump to one of the key points of what we found out of this particular study, and then I'll come back to it in a few minutes after I give you some other concepts to chew on. And the question was, are there any neighborhoods, or are there any cell types, particularly within neighborhoods, that are predictive of the outcome? So we couldn't look at just cell types alone and find any neighborhoods. We did find some any outcome relationships. We did find some neighborhoods that were predictive of outcome. But if you looked at particular cell types within certain neighborhoods, they in fact were predictive of outcome. So context matters. The spatial context matters. Just having a bunch of CD4 positive cells in your tumor is not as important as which neighborhood they're found within. So neighborhood 2 being obviously the main tumor. Here, it's okay, p-value is okay. The neighborhood 6, much better, right? These two as well, but none of them actually held a candle to one that we didn't quite understand at the beginning, but you'll see we've got a better sense of what it's doing now. When we published it, all we published was this association that PD-1 positive, CD4 positive cells, basically activated CD4 positive cells in neighborhood 9, were better for a diffuse inflammatory infiltrate cell patient than those who didn't have this cell. So what does neighborhood 9? Go down here and look at the key. Granulocyte enriched. So what is it then about granulocytes that when PD-1 cells are around them, what signals are they receiving that gives a better outcome? Now, is this cause or effect? We don't know. But this gets back to the original title, or at least the thing I talked about on the very first slide, is this is a new kind of way of thinking about targets, right? A diagnostic is implicitly a target, right? It might not be that close to the event that caused the diagnostic to be true for you, so that you could use it to predict an outcome, but somewhere back in the chain of causal events sits the initiating factor. So if these cell types are important for a good outcome in those patients, you have to ask the question, well, why does it not form in some of the other patients? What is there an event that's causing them to go away? Can I stop that from happening? Or did they never happen in the first place? Can I cause that to happen, right? Or can I make more of them? Is more better, right? So now, again, there's probably a molecular target that I would need to go after or tweak, or some process eventually. So at the end of the day, it'll still be a small molecule, or say a proteinaceous drug. But how we find it, it's not all about the omics of mass spec or a bunch of gene expression patterns, right? It's about thinking about how these cells are coming together in organizational units to achieve an outcome. So probably somewhere in there, and the way that we're going to actually approach this now is we're going to go back to those 750 patients, probably the 250 stage 3 and stage 4, pick out people all the way across the spectrum from the CLRs to the DIIs, create a pseudotime map of the patients that are at each stage, and see if we can find the Achilles heel. When is there a point at which these cell types, the PD-1 positive CD4s, are showing up in the, or going away from that cellular neighborhood 9, and are the cells that they're talking to are nearby, giving us a clue as to the event that we need to target to initiate, cause, or prevent from happening in the first place. So that's the roadmap that we're going to take in the next few years with these patients to figure that out. Now, what about other cancers? These are all the cancers that we're studying in the lab or have published on already with Paul Cavari, some of the squamous cell work. But I'm going to talk about the cutaneous T-cell lymphoma work of Darcy Phillips in the lab. So just like before, we had a clinical trial with only a small number of patients, because this is essentially an orphan disease, right? So cutaneous T-cell lymphoma, there's not many cases a year, but we were able to get a hold of patients in an anti-PD-1 therapy trial, responders and non-responders. This was enough. People think sometimes you need hundreds of patients, and it's the reflexive reviewer that you get back that says, oh, well, if you didn't do it with 100 patients and then you didn't do five blinded studies afterwards, the result is meaningless. Well, that's wrong. And that all really depends upon the density of the data that you're working with. And I think a lot of reviewers are accustomed to averaged information as opposed to the very rich data that we're able to discern out of the organization of how the immune system interacts with those tumors. All right, so like before, multiple cores per patient to sample the ecosystem. Here are the markers that we used throughout the study. A lot of functional markers as well here to look at the functional state. We mine them also with RNA at the same time. We have an adjacent section to do bulk RNA, and we move forward. So the disease has three stages. We have patients all the way across: the patch stage, early, plaque stage, middle, tumor, frank stage, frank tumor later. And you can see, obviously, there's changes to the architecture of the tumor. And of course, in this stage, there's more of the tumor cells, those red cells that you see scattered throughout. So we already know that the tumor microenvironment is a predictor of the stage and the therapeutic response, but what features are there that we can discern? So like before, there's the H&Es, there's the staining with key markers colored by the neighborhoods. We generally keep it to about 10 or so neighborhoods, and we name them with what we think of the primary cell types there, not the functional, but just the primary cell types as a way to keep track of things. We can break it down just like a fax into all the cell types that are there and then start doing some studies. So here's the epithelium. Across the bottom, you can see the cell types that are contributing to the epithelia here largely. There's a primary tumor dendritic cell neighborhood. Here's a tumor CD4 positive neighborhood in yellow. And again, all the patients had the same neighborhoods. Didn't matter whether we clustered individually on patients or we grouped all of them together, we ended up getting the same thing. So it's not driven by how we did the analysis. This is a feature of how the immune system organizes itself into functional units within these tumors. And then the rest. So let's just look at some of those organizations of cell types and how they change pre and post therapy in the responders, right? So here's the pre-therapy. There's fewer of these. Post-therapy, well, the therapy has initiated or allowed for the formation of more of those neighborhoods. That's a good thing, relatively. That's a frequency, not total, right? So there's more of those. Same for the CD4 tumor cells. Slight maybe enrichment for the Tregs. I don't think that's all that important. And then no change in the non-responders. So that's interesting, right? So non-responders, the therapy didn't change anything about the tumor microenvironment that was important for the responders. So that's nice. We did a whole bunch of statistics on this. We found some cellular neighborhoods, just like we found in the colorectal cancer. But basically the eagle eye of Darcy and Christian figured out that there was a rule that connected all of the observations under one umbrella, at least for the CTCL. And it's basically a spatial signature of response. And it actually makes sense. It's perfectly easy to understand. How close the tumor cell is to an immune effector, right? A CD4 positive or a CD8 positive cell, right? So how close it is to the immune effector versus how distant the effector cell is from an immunosuppressive cell. So if you add it all up across all the tissues, right? You can use the computer to basically find it all. You take then the division of A into B, right? Spatial score, A divided by B, a low value is predictive of pembro success, right? The closer the effector cell is to the tumor cell and the further away it is from these guys who are sending out negative signals, the better off you are. So here's the pre-treatment patients, right? Responder versus non-responder, all lower than the 0.5 score. For the responders, all higher here for those who go worse. And post-treatment, it actually tightens up and goes in this direction for the responders and goes in the other direction, more or less for the non-responder. So that's great, exactly as I think you would want it, and a simple score that you could use. We then actually worked with Martin Cheever's lab up in Washington with a smaller set of markers, only 8 markers that called out the main cell types that would give us activated immune effector cells versus suppressor cells versus the tumor. Redid the whole study with just those 8 rather than 56, and the study completely validated, meaning that you can go from a boil the ocean approach with antibodies at least to a subset of markers that predict the outcome for these patients, which is a great thing, obviously, for that. So, but we wanted to understand a little bit more then about the mechanism, and that was the purpose of the serial sections of the RNA. And it turns out that the responders were rich with markers that were somehow predictive or associated with the post-treatment biology, and a few in the non-responders. But this was what we were most interested in, understanding what the biology here is. That chemokine here played a significant role, we think, or plays a significant role. And really, there's multiple markers that all basically do the same thing in the responders. They go up, and these are, of course, inflammatory markers or markers that are sending out signals to the rest of the immune system to basically come hither. And little to no change for those same markers in the non-responders here. And then that's just some validation of that. And then we also looked at some of the basically the exhaustion markers, and they didn't change. Well, the TIGIT changed. I don't quite get this one and why it changes, but it does. But it's just the presence or absence of those exhaustion markers was reflective of the original response state of the patients themselves. And so for this part here, and I'm just calling out the PD-1 positive CD4 cells, the exhausted cells pre-treatment become effector cells post-treatment, apparently, or at least they're close enough to the event that are probably driving this. They change some of their expression of proteins, and they get further away from these negative cells as compared to the non-responders, which don't do much at all. So we're going to actually go back and in our other studies and look if this same phenotype can be seen elsewhere in some of the other clinical studies that we're working on. And as I said, working with Martin Cheever's lab, we were able to validate this with another set of markers. Okay, so now I'm going to take a step even further back and think about the immune system overall and the lymph node. There's about 220 or so different lymph nodes in your body. If you include the spleen and your other larger lymph nodes, and you were to look at them, you'd say, well, they're all lymph nodes. They all have germinal centers. They all have certain cell types, et cetera. Why bother? What's the difference? Well, they're all serving different functions, right? Your spleen is serving a different function than the lymph nodes under your arm versus the lymph nodes in your intestine. Are there common features of them? Are there common assembly structures of them that we could understand that tell us something about the local biology, and frankly, also tell us something about evolution? right? Are there conserved structures, which you would think of as being evolutionarily conserved? And then are there local structures which have iterated and evolved to serve local functions? So how do we begin this? So we start with the cellular neighborhoods, as I mentioned before. We're going to add another consideration here, and that is the interface between two cellular neighborhoods, which we call in this case, we're calling it a spatial context. Because as it turns out, when we started looking in these areas between two cellular neighborhoods, in fact, this is where there was a fair amount of variability, and there would even be varied between two different areas where, for instance, if there's a green and a red here and a green and a red here, depending upon the biology of where these things are or what they're being affected by in terms of pathogen states, the cells at the interface are different. So while the cellular neighborhood might be grossly called one cell type, it's the betwixt and the between where the fun actually happens. And of course, that's probably, it makes sense. If a cellular neighborhood is brought together by some sort of magnetic attraction of cell types, right, with their cell surface contact interactions, chemokines, secreted factors, et cetera, that are all somehow keeping the cells adjacent and near each other. There's another similar node of cells over here doing something similar. At that interface is where those two larger structures are talking. So that's where the variability is going to happen depending upon local biology, right? Or biological differences. So where and when these local biological processes interact and the signal propagation across all of these interactions is what we're going to start to schematicize next, again, automatically. So we're going to build neighborhoods of how the neighborhoods interact with each other and the interfaces that are seen or not seen, depending upon the presence or absence of these cellular neighborhoods. And this is all done automatically by programs written by two fantastic computational grad students in the lab. So here's an example of three different kinds of lymph nodes: the tonsil, a lymph node from under the arm, and the spleen. I don't know whether you call that a lymph node, but grossly put, it is. They all have germinal centers, but the germinal centers are different sizes, right? And although you'd call it a lymph node and they have a lot of the same things, there's obviously a lot of differences. So we start by creating the neighborhoods as before, right? And you can see, obviously, especially in the spleen, there are some major differences in terms of the macrophages that are there, the green over here. The germinal centers are actually quite different in their structure and their content here. And then you can basically assign the cell types to the neighborhoods and the presence or absence in different tonsils. Now, obviously, these tonsils are both of these tonsils were from patients who were they were taken out for basically throat infections. So there's probably some interesting biology going on that differs between those two. But so let's ask what the first level of let's say interaction is, and that's cell-cell interactions within a given neighborhood. So let's say we've got cells in neighborhood green versus cells in neighborhood blue. What cell types do the blue and the green like interacting with versus say brown and blue or brown and green? And so you can set up a map, for instance, like this. So here are all the green cells that only like talking to green cells or only next to. As you move towards the blue axis down here, you have more and more of these green cells that like to be near blue cells, et cetera, all the way down where you've got now only blue cells near blue cells, right? So one thing you can take away from this is that the blue part of the germinal center, right, likes interacting with the green part. It'll similarly like interacting with the B cells, but these cells rarely like interacting with those, right? So green over here is compartmentalized and kept away from the brown, right? So that's a feature of how these lymph nodes organize themselves, right? So this is all now done automatically. And then here's another one where you have, well, some of these cells are seeing just two of them or three of them down here. You can build out all these different structures and you end up with something that begins to look like this. Now, don't worry, I'm not going to take you through this, but this is the assembly rules for all 3 lymph node types that I showed you before. It's a composite of all of them, right? And the way that it's built is, here's a cellular neighborhood. That's the CNPI neighborhood. It likes interacting with the macrophage neighborhood here, or it will interact with this. And then it'll build another neighborhood nearby and another neighborhood. So these are all the possible arrangements of how they will organize. There's not all possible arrangements occur, right? So there is structure within the apparent randomness. Now, we can take that and we can look at, well, what's common across all the neighborhoods, across all the tissue types. So spleen, lymph node, and tonsil all have this structure where red T cells like interacting with the B T cell neighborhood, right? And you see it there, you see it here, et cetera, and you see it in the tonsil. But as it turns out, the neighborhoods that are similar are fewer across all of them, are fewer than those that are dissimilar. So lymph nodes have things that tonsils don't have, and tonsils have things that the spleen don't have, et cetera. So one of the next projects that we've got going is to actually get lymph nodes from a full cadaver dissection and to basically enumerate all of the different basically architectures that would be there. That's nice for an Atlas project. Why would you want to do that? Because cancers form in different parts of the body. And where's the first place they usually head to? The local lymph node. So understanding the local lymph node structure and why the cancer will take up residence in some lymph nodes versus others is probably hidden somewhere in how those cancers are able to use these rules to enable their own local growth. I'll give you some examples now. So back to the colorectal cancer, and hopefully soon to be finished. So again, remember, we had CLR patients and DIIs. We built out all these various neighborhoods with the codex. And now what we're doing is, like I showed you before, we're saying which neighborhoods are adjacent to other neighborhoods in all these different cancer patients, right? So here is the universe of organizations of what is allowed in this cancer patient versus this cancer, these cancer patients, right? So some of them are more structured and different than others, at least the image that we had for the section, right? So what's interesting about that, if you add all of them up, right, you take all of the sections that we had, right, the same ones that we published in the original paper, and you say, which of these certain assemblies are enriched in CLR versus enriched in DII? These are higher order assemblies, right? So 155 and 145, I'll show you those in a minute, are what are found more in the CLRs. And this particular neighborhood is more like, this particular set of assemblies of neighborhoods is more likely to be found in the DIIs. So here is the follicle. That's number 5. That's just an example of that. Here's 1 in 5 here. So that's when T cells come together with the follicle, right? So those are the colored neighborhoods. And we're coloring out to a range around the interface of about 5 to 10 cell depths on the assumption that cytokines or chemokines will traverse those number of cells and still have some reasonable effect. That's the reason why they're colored. So we're saying this region is a spatial context between the blue, in this case, the T cell enriched area, and brown, the follicular B cells, right? So that's the spatial context for there versus, and there's another one for the 3. And then this is more complex neighborhoods. In this particular patient, this is a DII patient where all 4 of these come together. And this includes specifically the tumor, right? So that's, again, this is still kind of an atlasing approach. So I mentioned that it's the cells at the interface that we're most interested in understanding how they might contribute to the biology of the disease. And so again, like before, we enumerate these and we build trees. And we're going to specifically pay attention to these markers here in this study because these are the activation cells and the activatable cells that we're interested in. And these are the activation markers, PD1, Ki67, and VISTA, that we're going to be interested in following up on, as I'll show you in a minute. And so just like before, we build these assemblies where the blocks and the colors of the blocks are telling us about the cellular neighborhood that define the interface, right? So the interface is, think of it as when this neighborhood comes together with this one, and that's the neighborhood. Call that the size of it, the percentage of cells that are belonging, the percentage of cells in those neighborhoods that belong to the spatial context at the interface. And we're going to color the markers according to whether they went up relative to the total tissue or down relative to the total tissue, to give us some indication of that there's something interesting happening at the interface that's not happening elsewhere, which would lend credence to the notion that the spatial context is driving local biology. That interface drives local biology. Okay, so here's the map of all of them, right? Again, I obviously can't take you through all of them. This is that map I just showed you before. Pay attention to the red and the blue. These are the ones I'm going to call up now. So here is basically something that happens specifically, or more so, I should say, in the CLR patients, right? These are the patients who do better. B cell neighborhoods come together with CD4 positive neighborhoods, and at the interface only, the B cells upregulate Ki67, so obviously they're dividing, and they've downregulated PD1. CD4 cells have upregulated PD1. Dendritic cells are also dividing, and they've downregulated VISTA, right? So PD1 and VISTA are both immune checkpoints, right? They're downregulated. So these are the patients who do better. There's no PD1 around. It's not turning off, presumably, the immune system, at least or these cells. So it's busy doing the right thing. Over here are the DII patients. They're doing something different. They're all basically running around trying to divide, but as it turns out, they're not doing anything functional. So we need to understand why the cells are dividing over here. But obviously, well, PD1 is up, so that's one indication of what's going on. PD1 is up on both of these cell types here, CD8s and the B cells. So obviously, this is contributing to a negative outcome, and on the on all three cell types, and VISTA in this case is down. So there's a lot of biology lurking in this map here, waiting for the right immunologist to look at it and say, eureka, voila, there's something here that needs to be understood. And again, remember, these maps are looking at just the cells at the interface, not the cells in the bulk neighborhoods, or at least it's colored according to the difference in those cells only at the neighborhoods, only at the interfaces. So here's just a one neighborhood to one neighborhood interaction in the CLR patients that are obviously statistically significant in terms of how these things interact, right? Here are the what we call the level 3 neighborhood interactions, right? And you can see that some which are found only in the DIIs versus only in the CLRs. That's interesting, yet to be understood. But if you look at CLR in these patients here, triangle 146, it's a T cell enriched, macrophage enriched tumor boundary. So the tumor is associated here. It's near the tumor boundary. And then DII is over here, this particular one, T cell enriched, immune infiltrated stroma, macrophage enriched. So the difference is near the tumor versus having infiltrated stroma. Both have T cell enriched, both have T cells and macrophages. So this biological difference is something that we're seeing. That is it caused by something else, or is it causal to the bad outcome? So turns out that this particular structure, and only when this structure is seen, right, where you've got macrophage enriched, as I said before, vascularized smooth muscle, T cell enriched, But next to bulk tumor, right, if you have this structure, you do poorly. If you don't have it, if you don't have the tumor nearby for these patients, maybe they've killed them, the DII patients do better, right? So this triangle is present in all of these different patients, right? Where in this patient, for instance, it's got in the CLR, it's got 3 of them versus the DII has 1 of them, right? In this patient, the CLR has only 1 of them, but there's many of these in the DII patients. That's how I basically say these are the patients that do poorly versus those that do well. And then these are all the other structures that can form in the relative numbers of them in the different patient types. This structure is equally as predictive as the structure that we published for in the prior Cell paper. Remember, I told you about the granulocyte enriched cells, right, and the PD-1 positive cells there. This structure is equally as predictive. So it's a corollary in some way, which we still have to understand, but the two together aren't any better because it turns out they're calling out the same patients. So there's another clue as to a therapeutic angle we can take. Can we make some of those structures come together or not? Could you take cells from a patient, pull them out, separate them, make them form in a, say, a gel, those neighborhoods, and put them back in? And is that now a functional unit, right? So you can maybe build these as therapies themselves, right? Can you force the structure in some sort of synthetic biology approach? I don't know, but something to think about. And then these are just some of the other neighborhoods that are there. But these are patients who don't have this particular motif, which in these are the patients who this is the motif I was telling you about before. This is the vascular, et cetera. These are the patients who do not have this at all, and they have other groupings. So it's almost as if you have this, you force certain structures here. If you don't have this, you force or you allow for other structures to form. So it's, if a signal is missing, for instance, formed by this triangle, other structures can form, right? So the immune system is always going to try to do something, but the presence or absence of certain structures will then cause other structures to initiate, form, and drive an outcome. And like before, you can do all of this analysis and basically build out these maps in many different ways. I obviously talked well beyond, and so I'm going to jump to the end and just thank the patients. There's another whole section of the talk. That stuff I just showed you about this tissue schematics is brand new, so it took me longer to go through. And I'll just jump to this final slide here. Just to the people in my laboratory who did the work. I've called them out here in gray. A lot of great technical efforts. I mentioned the effort of Martin Cheever before, and the validation study we were able to do with him. And thanks to our many funders, and thank you for your attention. And I'm sorry I went a little bit over there, but I think it's some exciting new stuff about how to think about how tissues are organized. And I'll stop there.
Fantastic, Garry. Thank you very much. That's really great. Questions? I guess I'll start with a question. There's a few coming through, but I'll just start. So I noticed, for example, that in the colon cancer study, you had a granulocyte enriched neighborhood. In the CTCL, you didn't have. And so I'm wondering, how similar or different are neighborhoods between different tumor types? And is there any benefit to trying to perhaps define neighborhoods in the same or similar ways in different tumor types?
Yeah, I mean, we saw actually very few neighborhoods that were similar across those two different tumor types. And perhaps that's driven by the fact that CTCL is a T-cell disease. It's a lymphoma, right? So it's probably driving a lot of the local biology of the immune system. The one thing that we did see, and I didn't really have time to touch upon, is, we ended up demarcating all of those lymph node neighborhoods, let's say, or lymph node structures, not so much neighborhoods, the way that cell types come together to form larger structures in what we call the tissue schematics. The basic tissue schematics we find in lymph nodes are found in both colorectal cancer and the CTCLs, right? So it doesn't matter where in a lymph node, where immune system cells are, they have rules that they use and how they come together. And there's certain ones that are going to be obvious, like T cells coming together with dendritic cells and et cetera. But there are higher order organizations of cells that are those transient arrangements of cell types coming together to do something, and then they go off and do something else that I was alluding to originally, that finding them in the lymph node is equivalent to finding them elsewhere in when they organize in a pathology to combat the disease. So not all of the things, obviously, that are found in the lymph node are found elsewhere, but many of them are.
Okay.
So that's,
okay.
To me, that's cool.
Right. Yeah. Question from Lauren Clubb. Do changes at the interface zones change the cellularity or phenotype of cells in the non-interface zones?
Good question. The interface, I mean, I suppose that there will be signals that are sent in. We don't have time series. Maybe the cells at the interface migrate in or migrate out. So I don't know. We haven't looked at that closely yet. And I suspect that this is where overlaying RNA is going to be important, right? Of looking at just because we see it with the protein, there's probably other events happening, say slightly more distally, that we're not picking up by the proteins because they're less responsive, if you will. And so the RNA work that we're doing, we have a new RNA spatial technology that I think will excite people.
Cool.
That we can see things down to basically subcellular across the whole thing, and it doesn't work with imaging.
Okay.
Coming by the end of the year.
You have to come back and talk about that one.
Mm-hmm.
Geoff Wahl has a question. Geoff, do you want to jump in? I think you can unmute yourself.
Right. Thanks very much, Garry. That was really beautiful and stimulating on so many different levels. I'm curious about whether you have looked at conditions such as pregnancy or obesity or other conditions that might affect cancer initiation or progression, and how this affects neighborhood structures, specifically, let's say, lymph node neighborhood.
Right. Yeah, great question. We have not segmented our populations according to obesity. Maybe we should go back to those colorectal patients and ask that question. Maybe there's a difference there. I suspect none of them were pregnant. But I don't know if you know Wendy Fantle, who was associated with my group. She's now got her own lab here at Stanford. She's been doing not pregnancy, but ovarian cancer.
Right.
And in ovarian cancer, she sees a particular kind of NK cell. It's called I think it's a decidual NK cell with particular markers that is highly upregulated normally at the placenta. There's many of them at the placenta, probably immunosuppressive. And in ovarian cancer, there's an extraordinary number of these same NK cells running around in ovarian cancer. So I think that's for instance, another example of a cancer taking advantage of local immune structures for its own purpose.
Yeah. I think the other thing that would be super interesting would be, what is the effect of chemotherapy? Right?
Yeah.
where, you're nuking a lot of immune cells and you get to restructure a lymph node in neighborhoods. And, of course, that can cause, in many cases response, but in many cases relapse also. And the disease relapses is worse. So is there some relationship to that? I think that's a great area for future investigation for you.
Yeah, even post the nuking of the immune system, the structures come back. They reassemble.
Are they the same or are they changed?
Yeah, at least at the level of organization that we're seeing, they're fragmented, as I said. They're there. I mean, all of the actually, you know what? You're right. All of those blocks were pre-treatment. So I don't know. You're right. Got to look.
Wonderful.
Gerald Churn has a few questions, may not get to all of them. Do lymph nodes have a neighborhood order that precedes disease?
Yes, is the answer. We've done this with melanoma in mice. And even a draining lymph node rearranges in let's say, expectation of the melanoma coming.
So you mean an animal with malignancy, but not yet any metastasis to the node?
Correct.
Cool.
Once it enters, all hell breaks loose. But there already is recognition prior to the metastasis apparently entering.
Right. That's interesting.
Probably a local inflammatory event somewhere. I mean, at the least. So
yeah.
The open question is the tumor preparing its bed for sleeping, right? For entry, or is the immune system trying to fight but being tricked to do the wrong thing?
Yeah, I imagine initially, the immune system is preparing to fight, but perhaps ultimately that gets subverted by the tumor to the benefit of the tumor.
Right.
I mean, it might not just be inflammatory, presumably. I mean, it could actually be migration of, for example, dendritic cells from the periphery to the nodes, for example.
Yeah.
Cell mediated.
I mean, that's always the problem with frequency and total cell counts. Capturing that multidimensionality in your head,
right,
is difficult.
Right. Gerald also wants to know what you found with UFO metals.
I can't tell you.
Secret?
Yeah, I think that is a kind of a side thing. I work with that guy, Lou Elizondo, and others who are all over the newspapers about the UFO stuff. They're friends.
Gerald says he saw you on TV.
Yeah.
Tony Hunter has a couple of questions. Tony, but I don't think you're able to unmute yourself because you're not a panelist. So if you type them in, we'll come to them. Pallav, go ahead. You got a question?
Yeah, Gary, this was amazing. I love your proposal to use synthetic biology to artificially create these neighborhoods. And I was wondering, do you have any clue what could potentially drive the attraction or repulsion of different cell types? I mean, in neurons, it could be, protocoherence or something, but.
Yeah, we don't know yet, but I suspect since these are mobile cells, it's going to be some kind of cytokines and surface markers. And again, I think it's going to be a combination of omics that are going to help us tease out what that local biology is. But you have to think of it as sort of like gravitational effect fields.
That's what I was thinking. Is it a field effect or is it contact based?
I think it's both field and contact. But I mean, overall, you could probably structure it. We should get some theoretical physicists on board who do quantum field structures. But that's how I think of it. That there's these local attractors.
Right, because if you could pinpoint those attractors, then you could use that could potentially be a target, right? Then you could drive these accumulations or neighborhoods in vivo. Right.
Yeah, I think it's a whole new area of therapy that we can get at. Just flip the diagnostic into, well, okay, somewhere behind that diagnostic is an event that is, eventually causal.
Yeah. Yeah, so then we need spatial methods that work in vivo, and we can get time resolved measurements.
Right.
Yeah.
So Murad Jaladi has a very important question, given that we're hoping to get this instrument at Stanford Burnham Previs. How standardized is the downstream computation analyses? Okay, so going from single cells to cell proximity studies and neighborhoods, is there a defined pipeline to be able to do this? Or
we've got them all up on GitHub.
Oh, okay.
Everything that we do is on GitHub. The latest stuff that we just have done on the schematics, I mean, we submitted it. It came back with some good, decent reviews. Fix this, clean up your language, and resubmit, and all of that will be provided.
Right. And that'll kind of identify neighborhoods de novo, or not,
Yes,
not just
absolutely.
neighborhoods that have been previously defined?
No, de novo.
Cool.
Yeah.
Okay. Great. I think, and there's one last question from Mengwei Ko is, actually no, well, yes, let me just do this one from Mengwei Tony, and then Tony's put his question in. Is there an NK cell neighborhood in colon cancer patients?
Is there an NK neighborhood? I mean, yeah, I think I showed one of them actually. That big blue one it has a lot of NKs there, but we didn't pay much attention to the biology of that. I can see Tony's question there.
Yeah, I was wondering the same. Do you want to read it out, Garry?
Yeah, so we're doing it 2D. Can you do 3D? Because it's funny, I just wrote, a new postdoc is going to be doing 3D, and I just wrote her letter this morning where I said, look, 2D is a pale reflection of 3D, and we're making a lot of assumptions about what's going on in 3D. I mean, probably the best person who's been doing this kind of work in 3D is Ron Germain, right? He's got 3D working beautifully with tissue clearing. And he and I worked a couple of years ago on getting codex working. He was using such thick sections, though, that it took a week to get the oligos in and out, right? And so I don't think whole lymph nodes are going to be able to be done, but I do think that we could probably do, let's say 10 cell widths. And we've been thinking about using some sort of electrophoretic approach to get the oligos and antibodies to kind of force them through rather than push them through with just liquid. We'll see, right? A lot of work to be done there on it. And what about different CAFs? As long as we've got the antibody markers to call them out, I mean, CAFs are, I don't know much about the differential biology of CAFs, but if there's antibodies, it's a, for some postdocs or grad students, there's probably a whole, set of papers to publish on defining them. I mean, we've been doing some work with stromal cells, and again, we call them stromal cells. I mean, come on, how many different kinds are there? And, and we all know that stromal cells can actually affect outcome in cancer already. A lot of work from Thea Tilsty on that specifically. She and I are working on that. They're using RNA to call out the biology that might be different. We'll hopefully get specific, protein markers, and then we can go back in with our stuff. Every time people say, well, why do you need 50 markers? Because somebody wants 51, right? Why do you need 51? Because somebody wants 60, right? So anyway.
Right. Tony also asked, sorry, go on.
No, that's good.
He asked about the spatial relationships. What do they represent in terms of cell diameters when you're defining?
So we only call out cells. The number for our cellular neighborhoods, we don't do cell width. We do cell number, right? So 10 cells out, not some average cell width.
Right.
If that answers the question. I'm sure that matters. I mean, big giant fat cells in the middle of something are going to basically act as an obstruction to certain kinds of signals and block certain kinds of signals.
Yeah.
There was a question up at the top there about, I saw.
Segmentation of lymph nodes.
Yeah, dense. Yeah, we don't have a problem with it. I mean, with antibodies, if you have enough antibodies overlapping, there's computational tricks to using an entire antibody set almost as one marker to call out the boundary of a cell. As long as the markers are not overlapping with a nearby cell, you can use 5 markers sort of as a dump gate, if you think in flow cytometry terms, to call out the locale of a cell. If you use just one marker, maybe it's only on one side of the cell and not on another. But if you use all the markers, you can better segment.
Right. And kind of a technical question then. So what's the limiting factor? At the moment you say you have hundreds of antibodies, oligo labeled and validated. What's the limiting factor in terms of, getting, thousands of antibodies available?
Number of NIH grants you can write.
No, I don't.
For it.
No, I don't mean to be able to buy that many and do, but for example, to have that many to choose from. Okay.
Oh, there's not. I mean, it's just, those are just the ones that we feel comfortable with and we validated. But you can go to AbCam, and there's lots of pathology validated reagents there. I mean, if you believe it. I mean, the first 4 or 5 years that we were doing this, the first pushback I always got was more antibodies equals more noise.
Right.
So, no, because this is a multidimensional space and noise is random.
Okay. And then Akoya will do the antibody conjugation to the oligos. Is that right?
Yeah, they have a custom service and they also have kits, I think.
Okay. Okay. Randy Kaufman has a question. How do murine reagents compare to human?
So as the target, I think there's fewer of the murine that we've validated, but that doesn't mean anything, right? I mean, Mark Davis, for instance, does a lot of work in mouse until he decided that mice were until he went on a rampage about mice not representing humans. But there's quite a few out there already. I mean, if it works usually in FACS, if it works in FACS, 9 times out of 10, it'll work in fresh frozen on mouse. It won't always work in FFPE, right? So there's like a Venn diagram of antibodies and how they overlap. We find on average 1 out of 3 antibodies work on FFPE. 9 out of 10 that work on FACS will work on fresh frozen. So if you can do it with fresh frozen, the world's your oyster.
Okay. Okay.
And fresh frozen means you can freeze it and then fix it. It doesn't need to stay wet. You can fix it and that'll stabilize it. It just means, don't put it in hot wax.
Right. Okay. Do we have any other questions? I think that's everything at the moment. If so, then thanks very much, Garry, for a wonderful talk. It's been really exciting to hear about.
Yeah.
And we all look forward to seeing more publications with it and hopefully getting to use it ourselves.
And Gerald, I'll tell you the answer over drinks sometime to your other question.
Okay, thanks, Garry.
Okay. Thanks, everybody.
Yeah, thanks all. July the 12th. And Garry's talk will be posted on the C3 Space Force website. Thanks, Garry.
Thank you.
You got a couple of nice dogs wandering around behind you as well.
Yes.
I don't know if you probably knew that they were wandering around.
Yeah, no, I don't think I can do about them. They rule the roost.
One of them came in with a ball at one point. I think he wanted you to play with him or her.
Yeah. Okay, well, thanks.
Okay,
everybody.
nice to meet you. Thank you.
Nice to meet you
Bye.
as well. Good luck, Pallav. Enjoy setting up your lab.
Thank you.